Hi, welcome to Neuroverse, a podcast hosted by Carolina
and Clara, where we discuss all matters from neuroscience to philosophy and beyond.
Welcome back to another episode of Neuroverse. Today we're going to be talking about theories of sleep and why we need to sleep. And we're joined by Raffaele Sarnataro, who is a postdoctoral researcher in neuroscience working in Gero Miesenböck's group at the Centre for Neural Circuits and Behaviour at the University of Oxford. Raffaele first obtained a bachelor's in biotechnology and then a master's in molecular and cellular biology in Italy before embarking on a PhD in neuroscience at the University of Oxford, where he first began researching the neurobiology of sleep, and during which he was also the president of Oxford University's famous Cortex Club, which is a neuroscience student forum that arranges debates, panel discussions, and symposia all around neuroscience. In 2024, Raffaele was awarded a Fulford Junior Research Fellowship by Somerville College in Oxford. And today, Raffaele will be helping us to unveil the mystery of why we need to sleep and sharing with us how fruit flies can help us answer this enigma. Welcome to the podcast.
Thank you, Clara and Carolina, for the invite.
Let's start off, as we usually do, by telling us a little bit about what brought you to study the neurobiology of sleep and about your research journey up until this point.
Yeah, so I've always been interested in sciences and life science, but my background, as you mentioned, is in molecular and cell biology. So that's what I trained in Italy during my undergraduate and master's. And I mostly worked in molecular and cell biology, but I've thought, even though the advancements in cell and molecular biology are indisputable, and I think we've witnessed decades or extremely successful discoveries that are also reflected in Nobel Prizes, for example, that go to discoveries coming from those subjects. I felt that there's only a certain level of questions that molecular and cell biology can answer. And I found neuroscience sort of transcending this level. Neurobiology is sort of an interesting bridge between molecular and cell biology and cognitive questions. And now we have the tools that allow to sort of enter this world. So I first actually got interested in neuroscience during my undergraduate when I heard a lecture talking about optogenetics, so it was a paper from Tonegawa's group where they implanted these light-inducible molecules that could activate neurons, and these basically allowed to trick animals in order for them to implant memories, really, so that they could be having fear reactions in contexts where they would not normally have, and vice versa. So this was, for me, absolutely revealing because it was almost magic. How could you, with the tools that molecular biology allow to create, be able to control the behavior of an animal? So it was an extremely elegant, I found, concept and also usage of optogenetics. And of course, when I was thinking of potential labs to do my PhD, this led me naturally to look into the person who actually first pioneered this technique, who is Gero Miesenböck, who became my PhD advisor. So he is based at the University of Oxford. And in the last years, he's actually not been working on optogenetics and tool developments per se, but actually has been using these and other tools to tackle other cognitive questions. And one of those, where the lab is mostly focusing on at the moment, is sleep. I found at first sleep quite uninteresting, and I actively didn't have much interest into it. But then, the more I entered the topic, the more I sort of found it fascinating, because it's one of the only behavior virtually for which we don't really know why animals display it. And the other interesting thing is that all animals have it. All animals that have been studied so far have it. For any behavior, of course, feeding, sexual behaviors, courtship, memory, it's obvious why animals do it. But for sleep, we know a lot, but we don't know why, or we have some hypotheses about it, why we need to sleep. And of course, there is much more to discover about how sleep is controlled in the brain. And this is what Miesenböck Lab has focused on, and also where I put my hands in. So this is the reason why I found this topic interesting. And I think we are now at the point of technology where we can tackle the understanding of the neural circuitry and the molecular basis of sleep well in many animals, including fruit flies. And the reason why fruit flies are interesting animals to study sleep is because if there is a function, a basic function for sleep, this means that it should be conserved throughout all animals, and therefore it makes sense to use one of the simplest animals that we can use to study this problem. And that's why Drosophila is interesting.
Yeah, I really like the emphasis on like neuroscience tools that got you interested in it. And relating this to Drosophila, I mean, I was reading your recent preprints that were talking about the complex techniques that you use in Drosophila to look at neural activity. And I just think like it's crazy that you can do, I work with mice. So to work at a scale that is so much smaller than that and be able to use very similar techniques is amazing to me. So I also know that a lot of previous research on sleep has been done in Drosophila. Could you tell us a little bit about what Drosophila has told us so far about sleep?
So Drosophila has been telling us a lot. So one model for which that people use to explain sleep and its control is the so-called two-process model. And this model postulates that there are two processes that control sleep. One is the circadian clock, and the other is the sleep homeostat. So the circadian clock basically entrains the animal behavior, but not only the behavior, also the function of many organs in the body, for example, to the dark and light cycle that are generated by the Earth rotation. And this is conserved throughout animals. And a Nobel Prize was awarded for the discovery of the genetic components of the core machinery that regulates the circadian clock. And this came from studies done in Drosophila. So the 2017 Nobel Prize was awarded for Drosophila researchers. So already one component of the process that controls sleep has been already deeply influenced by Drosophila research. The other component is the sleep homeostat. So sleep homeostat means it's a process that accumulates sleep pressure as the more time elapses since the animal has been awake, until this reaches a certain threshold and sleeping is initiated. Of course, if you deprive animals of sleep, then the area to integrate is larger, and therefore the following sleep is deeper. So normally after a night of sleep deprivation, for example, we sleep longer and more deeply. And in this, also for the sleep homeostat, Drosophila has been telling us a lot. More and more regions in the fly brain, for example, have been identified controlling sleep. And the advantage of finding these regions in Drosophila is that we can test a lot of hypotheses about the molecular underpinning of those regions, the plasticity of those regions, electrical activity, and we have the ability of target this neural population specifically up to the single neuron level. So we can understand how single neurons, single cells contribute to encoding sleep in different ways, or their activity, or their oscillatory activity, and so on and so forth. One important thing to mention is that, of course, sleep is a behavior. But of course, the way we think of sleep is a behavior, but is characterized by certain hallmarks that have been defined mostly from mammalian sleep. Now, the advantage of using another species is that these help us rethink what is the definition of sleep. What are the important criteria to define sleep behaviorally, but also in terms of, for example, electrical signatures of it and molecular substrates. So Drosophila has been telling us, and of course, the neurotransmitters, neurochemical, are conserved. Dopamine, for example, is a wake-inducing neurotransmitter as much as it is in mammals. But of course, the definition of sleep, the electrical activity the brain displays, the stages of sleep are a very much mammalian-centric definition of sleep. Now, in Drosophila, this has been quite rudimental until very recent, because the definition of sleep is very much behavioral. It's behaviorally defined. So locomotor inactivity, for example, so how much an animal has been immobile for is the current definition of sleep in flies. And of course, this seems very much rudimental compared to the elaborate electrophysiological recordings that mammalian and human researchers have to define sleep and different stages. But of course, the positive aspect of this is that this has also told us that we need better phenotyping of behavior to define sleep. So this has sort of pushed the field in the last few years to develop better tool to analyze different body parts of the animal, the different movements that these body parts have, in order and correlate them to different stages of sleep, arousability of the animal. And this has taught us a lot about how different animals display different behaviors, yet stereotyped when they fall asleep. And this also has revealed, of course, that the animals have different stages of sleep, that they enter sleep in different modes, behavioral modes, and they exit sleep in different modes. There are different kinds of sleep. The sleep deprivation, for example, affects the behavior differently depending on which kind of deprivation you use. This is only for a behavioral aspect. In terms of genetics, there is an incredible amount of detail that has been coming from Drosophila. Many brain regions have been identified more and more as controlling sleep. And the interesting thing is that many of these regions, of course, they display sleep-inducing or wake-inducing aspects, depending on which kind of stimulation you provide, which kind of subset of neurons you activate or inactivate. And I think this has been really inspirational for also thinking about how we challenge the neural populations, how we activate them, and what does this mean in terms of physiology. So with the ability and the toolkit that Drosophila allows, I think pushes the field to think deeper about the way we analyze and manipulate behaviors.
That's really interesting. It's amazing to hear how Drosophila has been such a good animal model to research sleep. And I find it really impressive how you're able to, I guess, see the effects of sleep in a single cell level. So I wanted to ask, firstly, because sleep, as you mentioned, is very different in Drosophila versus other animals like mammalian animals, what would be your definition of sleep? And also, could you clarify a little bit more how you can go into single cell resolution when looking at the effect of sleep?
I mean, I think sleep has got some broad features that are applied throughout different species. And of course, we have to bear in mind that each animal is different. So the ecology of each animal is different. There are animals, for example, that are able to forego sleep completely when the environmental conditions require it. For example, during migration, from one day to another, animal can completely forego sleep or change the way they sleep. Seals, for example, again, they can forego sleep, or they can change the electrical activity in their brains depending on whether they are foraging or not. So I think there is a lot of value also in studying different species in different ecological niches. Sleep in general is a behavior that implies quiescence. It is periodical, it is reversible, where the arousal thresholds are elevated, and that displays some homeostatic control. Homeostatic control means what I was saying before. So if you eliminate sleep or reduce sleep, then the following, when the animal is allowed to sleep afterwards, then it will display a more vigorous behavior afterwards. Reversibility, of course, but where the animal is asleep, its threshold to be aroused needs to be higher than when the animal is awake. So these are definitions that are applicable throughout all species. And of course, the operative definitions, they are different. So for example, in Drosophila, the first studies that looked into sleep were started in 2000. And those works, they tested that locomotor inactivity from about 5 minutes onwards could be called sleep for a series of reasons that imply this definition that I meant. Of course, now we know that in periods of immobility, for example, in a fly, correspond deeper stages of sleep, so where the animal is asleep for more than 5 minutes, they have some behavioral correlates. For example, proboscis pumping, the altere, so it's a part of their body that sort of moves downwards, and this is very stereotyped. So if we were able to identify these behaviors in a very high throughput way, then I think we will be better able to characterize and give an even better operative definition of sleep in flies. But of course, this depends on each animal. And in terms of the single cell resolution, Drosophila has got another incredible advantage. So its brain is complex enough. It's got around 150,000 neurons, but they're all very much stereotyped, which means that a neuron is always the same neuron identity. The number of neurons is roughly the same. And we have an amazing collection of driver lines that are able to label different parts of the brain, so different subsets of neurons. And now there's more and more genetic techniques that allow a finer and finer resolution of cells. So now we are really able, since a few years, to almost label each individual cell of the fly brain. This is another incredible advantage. And recently, there's two other more advantages that are, I think, absolutely amazing about Drosophila. The first one is the availability of connectomes. So there is not only one, there is several connectomes of the fly brain, and they basically provide the network wiring diagram of the entire fly brain. So we can know all the connection between all neurons to other neurons, and there is single cell transcriptomics data, many data sets of the whole fly brain, where we can match the identity of each of these cells, so the genetic makeup of these cells, to their structure and connections. And we even have individual driver lines that allow us to manipulate each of these cells. So we can really target any cell of interest and record from any cell of interest with multiple drivers, and we know the genetic makeup, which I think is fantastic.
Yeah, it's a true advantage to doing research with Drosophila.
Cool. So you mentioned how sleep may behaviorally present itself differently across species, but also that there could be a universal function of sleep. And as we know, why we need to sleep is not fully known yet. I'm really interested in the fact that sleep is not just a cognitive mechanism, but it may actually be important for the whole body in terms of restoring metabolic function. And I know that your research has focused on metabolism and its relation to sleep in the brain. Could you tell us a little bit about what you've discovered in terms of the role of mitochondria and sleep pressure?
So my research has been so far focused on trying to understand how sleep need is encoded in the brain. So basically, what's this sleep homeostat encoding or integrating over time? And to do that, we thought that a good way to start tackling the problem is to look at neurons that have an active role in controlling sleep. And our idea is that trying to understand how sleep is controlled will tell us something about the essential variables that are linked to the function of sleep. And what we discovered is that in the fly brain, a group of neurons that had already been pinpointed as be able to control sleep actually undergoes pretty interesting bioenergetic changes. Their mitochondria undergo fission and fusion. We can talk about it later. But basically, this determines the availability of ATP and therefore the electrical activity of these neurons, depending on the sleep history. So when the animals are sleep deprived, the mitochondria of these cells divide. This is a process called mitochondrial fission. And while they do so, they are also less able to produce ATP. At the same time, this, we believe, is a response to the increase in production of reactive oxygen species that the animal experiences when the animal is sleep deprived. And this is basically the homeostatic compensation that these neurons put in place. And in fact, when we artificially induce this compensation, the animals sleep less. And let's keep in mind, this is a population of about 12 neurons per hemisphere of an animal that's got 150,000. So if we manipulate the mitochondria of just this small population of neurons, if we divide them artificially, the animals sleep less, and they're also less able to catch up on sleep. And if we do the opposite, then the animal sleeps more, and they are also more able to catch up with sleep, and also the electrical activity of these neurons is higher. And we believe that this is because the ATP level and reactive oxygen species that are produced by the mitochondria in these cells change depending on the mitochondrial dynamics of these neurons. And in fact, when we directly control the ATP levels, for example, using a light-driven proton pump that with optogenetics is able to increase the proton motive force, producing more ATP, we actually induce sleep in the animals. And when we make these neurons express uncoupling proteins that decrease the proton motive force, then the animals sleep less. The interesting thing is that this was a sort of a lucky discovery, because we knew that these neurons were involved in sleep, we knew that these cells changed electrical properties, but we started tackling the problem with a very unbiased approach. I did single cell sequencing on these cells coming from animals that were either rested or sleep deprived, and we looked at what are the differentially expressed genes between these two different conditions. And it was interesting to see that there were two clear processes that were differentially regulated. So most of the genes that were upregulated in these cells were mitochondrial or metabolic genes. And this is why we sort of start looking at mitochondria. And the genes that were downregulated were synaptic genes. And this also led to another series of discoveries that we sort of continued in another manuscript. So we knew that these neurons had different reactive oxygen species level depending on the sleep history, but it was unclear where they were coming from, what was the mechanism that implemented it, and with this recent work, we sort of pinpoint to mitochondrial dynamics and ATP levels as a source of it. And I think the interesting point is also that this is a first mechanistic demonstration that aerobic metabolism is linked to sleep control. So now, again, we are looking at a small population of neurons, and the mitochondria in this population are able to sort of track the need for sleep and also implement it. And this is not a general feature of all neurons. And if you impose this manipulation in other cell types, this is not the case. So this means that these neurons do have a special role when it comes to metabolism, and they implement it through mitochondrial dynamics. Interestingly, this is sort of anti-cyclical to what the brain would normally do. So most of the theories that regard sleep and why we need to sleep postulate that we have some sort of synaptic potentiation when we are sleep deprived. And we actually saw the opposite in this population. And also people would imagine that the energy requirements during sleep are different, lower, or generally different, or more periodic. So the activity of neurons is more sort of stereotyped when the animal is asleep. But actually in this case we see the opposite. We see more energy being required by these neurons when the animal is sleep deprived. So probably this is where the relevance of these neurons for sleep comes from, the fact that they have an anti-cyclical behavior when it comes to controlling sleep compared to most neurons in the brain.
That's so interesting.
Yeah, it's so fascinating. And so I kind of wanted to probe out the causality of this and whether it's the mitochondria that's driving sleep or if it's the reactive oxygen species, and if we could potentially relieve oxidative stress by drinking electrolytes or eating antioxidant fruits.
Yeah, it's a very good question. So cell biologists, I think, have been working on this for decades. So the two phenomena generally go hand in hand. So what we showed is that the balance between the consumption and the production of ATP is what gives rise to the different reactive oxygen species level. And this is in turn what triggers mitochondrial dynamics in these neurons. And this is because when you have states where the ATP consumption is low, for example, in the case of these neurons, when the neurons are silent during sleep deprivation, because these neurons receive dopaminergic input, so the neurons are silent, they're less able to discharge ATP, and in fact, we measure that there is more ATP. In this situation like this, and the reserves of calories are full because the animals have been feeding, this puts the mitochondria in a mode that produces reactive oxygen species. And in fact, if this is extended over time, this triggers mitochondrial fission. And in fact, we have experiments that directly probe this causality. For example, we installed an alternative oxidase that comes from other species into the mitochondria of these neurons. So the alternative oxidase takes electrons from the electron transport chain and transfers them into water. And when you do that, basically you reduce the oxidative burden that is produced by mitochondria. When you do that, the fragmentation is rescued. And we also operated at the end of the electron transport chain, where ATP is produced. So we artificially consumed ATP using sort of stimulating the sodium-potassium pump. And also in that case, the mitochondrial fragmentation phenotype that we observed was rescued. So this shows that it's really the mode of operation of the mitochondria, the producer or not, the active oxygen species, that triggers these homeostatic responses. And then when we do it artificial, of course, we bypass the ROS levels. In general, though, I mean, this is a very interesting field in terms of just simply cellular biology. People have looked at the problem from both sides. They've artificially induced fission, and they've seen that the reactive oxygen species level change. And if you give drugs, for example, or compounds that are oxidants, then you produce fission in these neurons. Of course, you have cells are very different. This depends on their bioenergetic state, on the shape of their mitochondria. For example, mitochondria that are longer are more likely to undergo fission. So, of course, if you look at a cellular compartment or a cell type that's got longer mitochondria, you will be more likely to see a mitochondrial fission in that case. Now, whether this means that we have to to take electrolytes or antioxidants, I'm not sure. As a scientist, we've been looking at this problem in a very restricted neural population. So when you apply the same tools throughout the brain, this is not always the case, right? So, and the other thing that we have to keep in mind is that all the sort of peripheral organs feeding information. For example, muscle fatigue triggers higher need for sleep, but the brain is probably the only organ that needs sleep for its optimal function. And therefore, it's clear that all the animals that display a nervous system do display some kind of sleep. But we have to think that sleep is for the whole body, it's not just for the brain. So and whatever manipulation you do, of course, affects the whole body, and this will in turn affect sleep. So I think it's important to distinguish when it comes to sleep research what is sleep regulation and what is sleep control, and what are the sort of the system that in the body and brain feed sleep need informations to certain centers in the brain, and what actually actuates or puts in place the process of falling asleep.
I'm really intrigued by this specificity of the effects in this subpopulation of neurons. And I was wondering what makes them unique? Like you mentioned they get dopaminergic input. Is it because of the inputs they receive, or is it something more intrinsic, like their genetic makeup? Or are their mitochondrial DNA different, or anything else? Like, I'm sure it's a combination of things, but what do you think is really contributing to this specificity in the population?
Yeah, it's a very good question. I think still, I think a lot of research needs to be done on this, but I think it's two components. One is their circuitry, so that they receive exactly dopaminergic inputs that will hyperpolarize these neurons when they receive awakening stimuli. And the other thing is the fact that independent groups have shown that these neurons, they have a high mitochondrial turnover and a high level of oxphosgenes. So it's possible that these neurons are really tuned to a very high level of mitochondrial activity. So they're more sensitive than others in sort of picking up metabolic and nutritional information. And the combination of these with the ability to discharge or no ATP coming from the circuitry, in particular from dopamine, creates sort of this mismatch that is used by the brain to track sleep. Now, I don't expect these neurons to be the only centers in the brain that control sleep. As I told you before, there's evidence in flies that there's multiple brain regions that control sleep, even though different regions have been studied with different level of detail. And it's possible that the different brain regions or neural population control different aspects of sleep. I mean, sleep is a really, as I told you before, it's got many features. There is the homeostatic component of sleep, which is probably these neurons that I've been studying mostly control. There is the arousability threshold. There is the duration of sleep, the architecture of sleep itself, the gating of visual inputs. These are all features that are absolutely important. The coordination of these features is what actually allows the animal to naturally fall asleep. And of course, this is true in mammals even more. In mammalian brains, there have been plenty of regions that have been identified as sleep-inducing.
So I really liked how in your paper you do mention a little bit about the history of aerobic metabolism and how it was essentially a revolution that allowed eukaryotes to maximize free energy yield. So I have a bit of an out there question, which is, do you think that if we evolved to carry out anaerobic respiration, we would still need to sleep, if at all?
I mean, this is a speculative hypothesis.
Of
I think the
course.
interesting bit is that we see sleep as soon as the first nervous system appear. And I think that the main property of nervous system is that they are plastic and that they are non-renewable. So I think the combination of these two is what somehow requires a state that is sort of solidified into sleep. And of course, this has taken other functions. They sort of fed into it: immune function, restorative function, clearance, and so on and so forth. But probably this is the key aspect that we have to remember, that sleep is for the brain, and the brain is special in this regard. Of course, neurons are very energetic demanding cells, but so are muscles, for example. And the main difference is that they are not renewable. And the other thing is that, of course, the main function of the nervous system is to be plastic. And in fact, one of the most influential hypotheses on why we need to sleep is the so-called synaptic homeostatic hypothesis. So according to this hypothesis, while we're awake, our brains experience things in the world, and therefore the connection between our neurons strengthen. And of course, if this continues, the signal-to-noise ratio of information transmission in our brain would become poorer and poorer. So there is need for a moment of disconnection from the external world when our brain can sort of scale down overall synaptic weights and just keep the connections that are relevant, for example, in terms of memory, but overall downscale synaptic strengths. This has been proposed first almost 30 years ago by Chiara Cirelli and Giulio Antononi, working in University of Wisconsin-Madison at the moment. And there have been many studies from their group and others showing that actually this seems to be the case at the ultrastructural level, molecular level, in different animal species. So it seems that there is an important property is actually specific to neurons, which are synaptic connections, that does change quite dramatically with sleep. It's not in all the neurons, it's not throughout the brain, but overall, this seems to be true at many levels.
I have two questions about cross-species differences related to that. The first is because you mentioned all animals with a nervous system sleep. So do you require a nervous system to exhibit sleep? And also, does your metabolic rate of an animal correlate with how much it sleeps or anything to do with sleep behavior?
So I would say that sleep is mostly a behavior. Of course, there are properties of sleep, for example, oscillations, certain kind of oscillations that you can display in a dish, and people have worked on that. In mammalian slices, for example, there are certain kinds of rhythm that are sort of entrainable or consistent if you look at slices, organotypic slices or acute slices of different brain regions. In flies, the same. There's a lot of research done on rhythms that you can identify in ex vivo explants that change depending on the time of the day the explant has been taken. But I would say that sleep is mostly it's a behavior. So these are electrical signature of sleep that, of course, are extremely informative. They can tell us a lot. There are interesting correlations between metabolic or body parameters and the amount of sleep that an animal displays. For example, and this is work mostly carried out by Geoffrey West and Van Savage, that looked at the size of the brain of different mammals, in this case, and the fraction of their daily time spent asleep. And there is an interesting relationship between those, meaning that the animals that have a smaller brain tend to sleep much more than animals that have a bigger brain. But the correlation is interesting because in one of their main papers, they show that this correlation takes the shape of, or could be described by a power law, and the power law exponent is a multiple of 1/4. And the reason why they speculate is 1/4, that is because, and not a multiple of 1/3, which we would expect from Euclidean scaling, is because the other dimension are the vascular distribution network within the animals. They distribute resources, in this case oxygen, for example, within the animals. And these power law multiples of 1/4 are very common when you look at these kind of correlations. For example, body mass, metabolic rate and body mass, and so on and so forth. So this hypothesis of redistribution network seems to be sort of a major driver of evolution under many aspects, and one of them is sleep.
Wait, so if you have a smaller brain, you sleep more.
Yes.
And how does that relate to metabolism?
So the idea is that since the way you correlate the size of the brain to the amount of the animal is asleep is via a power law, which has the exponent of 1/4. This is an extremely common feature between different sort of metabolic rates and anatomical features. And this is because the quarter, so the fact that there is a fourth implies that there is another dimension that is given by distribution networks. And one of these could be capillaries, for example. Of course, this is speculation, but I think it's a really interesting correlation that you can find throughout all the mammals for not only sleep, but many other body parameters.
Right, okay. But that's also interesting because if you link that to what you're saying about synaptic homeostasis, then you'd expect that you would need more sleep if you had more synapses to homeostatically bring back to normality. I know the synaptic scaling hypothesis is a hypothesis, but yeah, how do you think that might fit together?
This is an excellent question. I think putting together so many evidences, I think, is really the challenge, because this is one of the theories of why we want to sleep. There is clearance, there is the energetic hypothesis of sleep. There's many. I think the key is in looking at the sort of the electrical reorganization of the brain and what's the biochemistry that allows it or that is prescribed by certain electrical features. For example, when mammals fall asleep during non-REM sleep, you see certain kinds of oscillations, slow wave activity mostly. Is this just an epiphenomenon of sleep, or inducing it per se has got some beneficial properties just for the cortex or for the whole brain? I mean, we are starting working on this. Now you can manipulate large portions of the brain to sort of mimic these oscillations, but we are really just at the beginning. So I think I agree with you. There's lots of hypotheses and lots of possibilities, sort of integrating them together, understanding how they fit with each other. I think this is really the challenge of sleep. But this has been historically the case. I think the problem of localizing sleep and understanding where the need for sleep comes from, it's old. Already more than a century ago, Konstantin von Economou was the first one to sort of propose that the sleep is a location problem. And he sort of started looking into this because he was a doctor, and he had patients that had a disease called encephalitis lethargica. Encephalitis because it was a brain infection, lethargica because some of the patients were hypersomniac, and some of them were actually insomniac. And they found, looking at the brains of these patients, that the problems resided in the place in the brain where the infection had hit the most. And he saw that there was sort of a boundary between the midbrain and the encephalon. And if lesions were on one side of this transition boundary, then the patients would be insomniac. And if we're on the other side, then they would be hypersomniac. And many others, Giuseppe Moruzzi and others, for example, afterwards, sort of rationalize the problem in a formal way that the sleep is a property of the brain, but you need some centers that control it, that regulate it. Otherwise, if each neuron will sleep by itself, or each neuropopulation will sleep by itself, the brain will be in a so-called, Giuseppe Moruzzi writes, dormiveglia, so half sleep the whole time. You need some centers that coordinate this activity throughout the brain. And therefore, this means that there are regions of the brain that sort of execute sleep functions. And this could be, for example, downscaling of synapses. And other brain regions sort of track the need for sleep and they impose it on the rest of the brain.
Which is what you showed as one example. But since this area that you speak of in Drosophila, the changes there are so specific to that area, do you think there is like a mammalian equivalent? I know this is also speculative, but I know there are also specialized areas in mammalian brains for sleep control. But do you think that maybe a similar mechanism may be happening, or could there be completely different neurobiological mechanisms?
I think there is, so there are neurons in the mammalian brain that are sort of analogous to the neurons that we've been studying. In particular, neurons in the ventrolateral preoptic area of the mammalian hypothalamus have a lot in common with these neurons. They are inhibitory, they are peptidergic, and the receptor of the peptide they produce are orthologous between the two. They are sleep active. They respond similarly to anesthetics, for example, so they could be similar under many points of view. Although another project that we've been working on, which is mostly a collaboration between me and another postdoc in the lab, showed that these neurons, the same neurons that we were talking about, where we now know what causes changes in the excitability levels, display oscillations too. And they display oscillations that are in a very, very similar range to what the mammalian cortex displays. They are different because in the flies, the intensity, so the power of this oscillation changes with sleep need, but they persist throughout their vigilant states. in mammals, this is not the case. You find mostly slow wave oscillations where the animal are in non-REM sleep. So these neurons can be homologous to cortex, if you wish, in terms of execution, and to hypothalamus in terms of sensing. Hypothalamus is one of the neurosecretory regions that is most important for controlling hormone release and for integrating sort of feeding needs and other sort of basic functions. So it could be that in the fly brain, it's much more compact and compacts together sort of many functions into a smaller population of neurons.
Yeah, so interesting.
So, going back a little bit to the broader picture in terms of oxidative stress and sleep, you also mentioned in the paper that sleep, like aging, may be an inescapable consequence of aerobic metabolism. Could you speak a little bit about the relationship with the two? And I guess going back to the crux of it in terms of whether sleep and mitochondria is really dependent on oxidative stress or not.
Yeah, so oxidative stress happens in all the cells. Of course, neurons are much more sort of energetically demanding. As we were saying before, they are not renewable. Therefore, they are more sort of prone to damage. And this is thought to be one of the reasons why there is a decline in neuron functions. You have peroxidation of lipids, for example, and of course, this all cascades to problems in repair and so on and so forth. And this happens throughout all the tissues, really.
Do you think sleep and aging go hand in hand?
So it's been shown in several animals and several species that with sleep deprivation, you do have either increase of oxidative stress in certain brain regions in different animals, and you have damage of certain structure. And of course, this could lead to extreme consequences. Even I think sleep deprivation for 5-6 days has been shown to lead to pro-apoptotic markers increasing in the hippocampus. So definitely oxidative stress and sleep are linked. Now, oxidative stress also increases during aging, and there's plenty of evidence that shows it now. Whether this is a property of only the brain or all the cells, I think the latter is much more likely. So that all the cells experience it, and this leads to many consequences that we call cell senescence.
Would potentially, because I think many people talking about sleep and how, sleeping more and lots can help reduce aging or at least the speed of it. Do you think that as a theory makes sense in terms of what we know about the metabolism?
Yeah, absolutely. I mean, there is undoubtedly a lot of evidence that shows that sleeping little has got negative consequences for the brain, for memory. I mean, one of the main hallmarks of sleep loss is the inability to remember. I mean, this is true even in flies. And of course, the cognitive impairments of loss of sleep are incredible. In humans, for example, when there is the time shifts between summertime and wintertime, for example, there are more accidents, more heart attacks, because people don't have enough time to catch up, or their circadian clock is mismatched with their sleep homeostat. So there's a lot of evidence that shows that there is cognitive impairment and heart failures and negative consequences for the brain, for the body and brain, if you sleep less. So definitely a good night of sleep and having regular schedules is definitely going to slow down the cognitive impairments or any negative consequences for the body. Immune functions are impaired when you lose sleep, for example. This is another very evident cause. The likelihood of catching a cold, if you're sleep deprived, is higher than if you're not. The likelihood of getting a efficient vaccination, if you were sleep deprived before getting the vaccine, is lower than if you had a good night of sleep, for example. So there is plenty of evidence. So definitely a good night of sleep and having regular, clean sleep hygiene is absolutely beneficial for brain and body.
Yeah. And I was wondering, in the experiments that you did with Drosophila, you mentioned that you would sometimes induce fission of mitochondria. I don't know if you did behavioral experiments, in terms of, would you notice whether the cognitive functions or behavioral functions of the Drosophila were similar when they were either naturally sleep deprived or mitochondrially induced physiological sleep deprivation in that sense?
So that's a great question. So we mostly looked at sleep and arousal threshold. These are memory and lifespan, I think, are the two obvious sort of follow-up experiments that one could look at. And it's research that is actually actively being done in the lab at the moment. Anecdotally, people have tried to promote fission in the whole Drosophila, for example, and this increases lifespan, apparently. But of course, these are crude manipulations, right? So then, and the other thing is that one has to induce fission to make sure that you are inducing it, because the core machinery that we play with is extremely complex. So it's regulated by a lot of post-translational modifications. So if you induce fission, you always have to make sure that this induction is consistent throughout the lifespan and there's no compensatory effects. But anecdotally, people have seen that if you induce mitochondrial fission, it prolongs lifespan. But these are absolutely important questions.
I guess if we were to relate it to real life in a way, I guess essentially like what is the difference between like natural sleep, like going to sleep, and in a way induced sleep, like taking melatonin, for example, and whether you are able to get all of the benefits from sleep if it's induced in a similar way that with like mitochondrial fission, you're manipulating it.
Yeah, so far, I mean, there's no drug that is able to sort of fulfill all, so make you fall asleep and fulfill all the benefits of sleep. The only therapy that is actually been approved in this sense is a cognitive behavioral therapy, where if you're insomniac, for example, you reduce your sleep schedule. So you try to go to bed later at night, as late as possible, and wake up early, and sort of slowly increasing the time that you spend in bed. And also you have a better sleep hygiene in terms of heating habits and environment and so on and so forth. But this is the only treatment for insomnia that we have at the moment. There's no pill that is able to target. This could be because many of the findings that we've been talking about are really in flies, but also in humans or in mammals in general. They've been focused on certain brain regions. So we know a lot about the biochemistry and cellular biology and the neural activity of certain brain regions, which are generally key controllers of these behaviors. But target them specifically, of course, is much more challenging. And if we talk about oxidants, there is also a good role. So oxidative stress is also a signaling mechanism that the organism does use for signaling in general. The cells normally use for signaling. So the reactive oxygen species are not necessarily a negative thing for cells. So I think all these claims about reducing your ROS levels are a bit too strong. I think they're strong claims. Redox signaling is a natural signaling for the brain use. Now, of course, integrated over the course of time can lead to problems, but this is how our cells are built.
Just switching gears slightly for a second, I wanted to ask about the oscillation work that you mentioned. And first of all, because you mentioned brain specificity again, and we've spoken about it a lot, but were the oscillations observed specific again to the same brain region, or has it been shown in Drosophila that there are brain-wide oscillations? Because I know in humans and other mammals that oscillations can be picked up from the cortical surface and they are quite extensive throughout the brain.
Yes, you're right. In mammals, the sort of hallmark oscillations of sleep are cortical and they are very broad. In flies, there is work done years ago that's shown that you can pick up sleep-specific oscillations if you look in the central complex, which is basically sort of an integrative center, something that you could put in analogy with the cortex. If you put an electrode there called the LFP, then you can find oscillations that are specific to different sleep stages. So these are LFP recordings. The kind of recordings we've done were mostly calcium recordings, mostly. But we and also other groups have shown that different components of this central complex, to which the neurons that we've been studying belong to as well, do display oscillations. It's not just these neurons, there's other kinds of neurons, they display oscillations, and they are in mutual contact. So there is work that shows that all these neural populations that display oscillations, they talk to each other. So it really seems that the central complex of flies includes certain kinds of populations that they oscillate at different rhythms, and that together they create some sort of collective oscillatory activity that can be picked up by LFP recordings. And this could be analogous to what happens in the cortex with the corticothalamical circuits, for example.
Yeah, and that kind of links to my second question on that. That was about causality, and perhaps you could link this also to the mitochondrial findings, but in terms of how activity is related to the more biochemical basis of everything, do these oscillations, do you think they have a function in inducing the biochemical changes, or are they an outcome of it?
Yeah, absolutely. So the work on the circuitry of these neurons, I think, has been very revealing, because it's shown, one, that there is mutual connections between these sleep-inducing neurons that we've been talking about and dopaminergic neurons. They inhibit each other. We discovered how they inhibit each other, and that Dopaminergic neurons, of course, release dopamine on these neurons. Once they release dopamine, these neurons become silent. And of course, if you imagine a sleep deprivation paradigm, or if the fly, for some reason, ecologically needs to sleep less, then you could imagine that this release of dopamine is sort of consistent or more tonic over time. And this sort of silence the neurons and puts them in a mode that allows them to undergo fission, reactive oxygen species, more ATP being stored in the cells, and therefore all these biochemical processes that sort of put the neurons in a state that are more excitable once the system allows them to display their electrical activity. So when they are not as inhibited by dopamine, they're more excitable. And this excitability is then implemented with this pattern of oscillations. We've shown that these neurons actually display oscillations in a slow wave rhythm, and that the power of this oscillation increases. And these oscillations rely on a mutual inhibitions between two different hemispheres of the fly. So these neurons talk to each other, they inhibit each other, and they're more excitable after sleep derivation. So this mutual inhibition is what creates these increased oscillations. And actually, if you impede this mutual inhibition, the oscillations are dampened. We've artificially even replayed these oscillations with optogenetics. So we have activated these neurons with a certain frequency, and if this frequency was actually displayed in bursts that mimic the strong power of these slow wave oscillations, then you sleep much more than if this induction was tonic. If this induction was tonic, then the animal either did not sleep more or the induction was little. But if the activation was done in simulation patterns that mimic the burst of the slow wave activity, then the induction of sleep was significantly higher. So this shows causality. These oscillations that come from different excitability of these neurons, they're actually causative to sleep. And if you, again, impede them, then sleep is impaired. The other interesting thing that we discovered is that there is homeostatic plasticity. Again, these were findings that came from this single-cell transcriptomics that was unexpected. And it looks like from modeling in a system where you have recurrent circuitry and neurons that increase the excitability upon sleep deprivation, that you need synaptic depression in order for this oscillation not to be completely dampened by this system. So this sort of explains why we've seen this paradoxical effect to what you would expect in the brain.
Yeah, it's really so complex. Like while you were describing how the changes at the biochemical level may influence the activity and the oscillations, I was also then thinking about synapses. And then when you mentioned that, I was like, okay, it puts it all together, but in a really complex way where everything is influencing everything and it's like cycles and cycles.
Yeah,
But
that's
yeah,
the beauty of Drosophila, that you can sort of study different mechanisms within one cell, as we were saying at the beginning.
yeah, fascinating. Cool. So I think we'll wrap up today's episode there. Thank you again so much, Raffaele, for joining us. And it's amazing from somebody who wasn't initially interested in sleep to now being an expert in it and being able to apply your fine-tuned knowledge of these biochemical processes to the bigger picture. So thank you so much.
Thank
Yeah.
you.
Thank you, Raffaele. It's been so insightful, and thanks for answering all of my probing questions.
Thank you for the interesting questions.
And thank you for listening to Neuroverse. If you enjoyed today's episode, share it with your friends and family, and be sure to check us out on social media at neuroverse_pod. And for more information, check out our website, neuroversepod.com.